Good morning, dear students. This is your Sudha ma'am. Welcome to the third session of lesson 4, that is excretion, the wastage disposing system. In our last session, we have learnt about excretory system in humans and internal structure of kidneys. In this session, we are going to study about mechanism of urine formation and micturition. Before starting that, let's recapitulate the structure of nephron, which is the structural and functional unit of kidneys. Typically, a nephron consists of two parts, that is Malpighian body and renal tubule. Malpighian body further divides into a cup-shaped Bowman's capsule and a glomerulus. A glomerulus means nothing but a bunch of blood capillaries. And the Bowman's capsule is single layered structure and it is made up of squamous epithelial cells called podocyte cells, and in between these podocyte cells pores are there which allow the glomerular filtrate to enter into the Bowman's cavity. And renal tubule is divided into three parts, that is PCT, first or proximal convoluted tubule, second one loop of Henle, which is a U-shaped structure, and the third one is DCT, second convoluted tubule or distal convoluted tubule. First one proximal, as it is close to the Bowman's capsule, we will call it as proximal convoluted tubule, and DCT, as it is distant from the Bowman's capsule, it is DCT, distal convoluted tubule. And the renal artery divides many times and forms efferent arteriole. These efferent arterioles enter into the Bowman's capsule and forms the glomerulus and comes out as efferent arteriole. This efferent arteriole divides into many capillaries and surrounds the PCT, loop of Henle and DCT, and called peritubular, and forms peritubular network. These peritubular capillaries joins together and forms renal venule, which join with other venules and form renal vein. This is briefly about the structure of nephron. Now, let's move on to the mechanism of urine formation. The formation of urine involves four stages. First one, glomerular filtration. Second one, tubular reabsorption. Third one, tubular secretion. And fourth one, concentration of urine. These are the four stages in the formation of mechanism of urine formation. The first one, let us discuss about the first one, that is glomerular filtration. Blood flows from renal artery to efferent arterioles. This efferent arteriole divides into many capillaries called glomerulus and again united and reunited into efferent arteriole. Already we discussed this point. And the diameter of efferent arteriole is narrower than the efferent arteriole. Efferent means the arteriole which is entering into the Bowman's capsule. So, the diameter of efferent arteriole is narrower when compared to efferent arteriole. So, this exerts pressure in the glomerulus and filtration takes place, what we call it as ultrafiltration. Due to this pressure, the blood is filtered in the glomerulus. Waste molecules and some useful molecules and large amount of water are filtered out and enter into the cavity of Bowman's capsule. The lining of the Bowman's capsule is formed by podocyte cells which have pores. So, this allows the filtrate to enter into the cavity of Bowman's capsule, and the thick blood, that means which is not filtered, which contain more solid particles and other substances, the thick blood passes into peritubular capillaries through efferent arteriole. That means the blood which is filtered through glomerulus enters into the cavity of Bowman's capsule and it passes through the PCT, loop of Henle and DCT, and the blood which is not filtered that is somewhat thick also, that enters into the efferent arteriole, peritubular network through efferent arteriole. Now coming to the next process, that is tubular reabsorption. Filtrate from glomerulus is called primary urine. Whatever the filtrate we got, that is called primary urine, and this is different from the urine which generally we excrete because it contains many useful substances and large amount of water. The chemical composition of primary urine is almost equal to the chemical composition of blood except the presence of blood cells. Only blood cells are not get filtered, remaining all like glucose, all useful things let us discuss. So it is equal to the blood, hence we call it as primary urine. It is different from normal urine which generally the human beings excrete. So the primary urine from the cavity of Bowman's capsule passes into PCT, that is proximal convoluted tubule. Here the useful substances in the primary urine like glucose, amino acids, vitamin C, ions of potassium, calcium, sodium, chlorides and 75% of water are reabsorbed into peritubular capillaries. So all these useful materials are again reabsorbed into the peritubular capillaries from the PCT to peritubular capillaries and finally carried to the renal vein. That means they are re-entered into the bloodstream. That means they are not get wasted, they are re-entered into the bloodstream because they are useful substances to our body. This tubular reabsorption takes place in the PCT, that is proximal convoluted tubule of renal tubule. And the next process is tubular secretion. After reabsorption in the PCT region, the primary urine travels through the loop of Henle onto the DCT. Nearly 10% of water is reabsorbed into the peritubular capillaries in the loop of Henle region by osmosis. As it is travelling through the loop of Henle, the water is reabsorbed. Almost 10% of water is reabsorbed into the loop of Henle region and by osmosis. Now the filtrate passes into the distal convoluted tubule where the tubular secretion takes place. It is active secretion, that means it requires some energy. It is the active some energy is utilized. It is the active secretion of waste products by peritubular blood capillaries into the DCT. So here the secretion of waste products, still if there is any waste products in the blood, from the peritubular blood capillaries to the DCT. It ensures the removal of all waste products like urea, uric acid, creatinine and salts, ions like potassium, sodium, hydrogen, etc. from the blood, and this maintain proper concentration and pH of the urine. So in DCT region, the materials which are not useful to the body like urea, uric acid, etc., they are secreted from peritubular blood capillaries to DCT. And in PCT region also some amount of tubular secretion takes place. And next stage is concentration of urine. So, already 75% of water from the primary urine is reabsorbed in PCT and 10% of water is reabsorbed in the loop of Henle. Further, concentration of urine is regulated by a hormone called vasopressin. Vasopressin or ADH. We will call it as ADH, that is antidiuretic hormone. It is secreted by the posterior part of the pituitary gland. Our master gland, pituitary gland is there, no? So, from it is the secretion of pituitary gland and it is called antidiuretic hormone. Anti means opposite, diuretic means removal of water from the body. Removal of water from the body is called diuretic. So, this is opposite of removal of water. That means this hormone do not allow the body to lose water. Vasopressin helps in reabsorption of water in the renal tubule. The main function of this vasopressin hormone is reabsorption of water. It helps in the reabsorption of water in the renal tubule so that it maintains the osmotic concentration of the body. That means it maintains the fluid balance and the concentration of electrolytes. If a person drinks excess water, the osmoregulation is maintained externally and excessive reabsorption of water from the renal tubule is not required. Sufficient water is there in the body, then there is no reabsorption of water is required. Thus, vasopressin, in such conditions, vasopressin is not secreted. But if the water content in the body is less and still if we are losing water through urine, then osmoregulation is not maintained and dehydration takes place, isn't it? To prevent such conditions, pituitary gland releases vasopressin. So, more water is reabsorbed into the kidney tubules. We consume lot of water in summer, but still we urinate fewer times in summer when compared to winters. Why? This is because we lose lot of water from our body through sweating in summer. Generally, we lose water through sweating. So, to maintain the body fluid balance in the body, vasopressin is released. As we are losing so much of water, so we have to maintain this fluid balance, fluid and ion balance. So, that is the reason vasopressin is released. Whenever vasopressin is released, our kidneys reabsorb more water. So, we urinate fewer times in summer and the concentration of urine concentrated urine is passed out. And the deficiency of vasopressin leads to the disease called diabetes insipidus. Diabetes insipidus. And the symptoms are excessive, repeated and dilute urination. A person who is suffering from diabetes insipidus, generally they passes excessive, repeated times and dilute urine. Okay, and this is not the normal diabetes. Generally we will listen, no, diabetes, sugar disease, something like. So that is diabetes mellitus and it is caused due to the deficiency of insulin in our body. That is different. This is diabetes insipidus and it occurs when the body cannot regulate how to handle the fluids and it is marked by the intensive thirst and heavy urination. Moving on to the next topic, that is micturition. Release of urine from the urinary bladder to the outside of the body is called micturition or urination. That means release of urine from the body is called micturition. The urine is temporarily stored in the bladder and there are two sets of circular sphincter muscles in the bladder. Both the sphincter muscles are constricted and close the exit of the bladder when it is filling up. The ureters bring the urine from the kidneys to the urinary bladder When it is filling up, both the sphincters are closed. But as the volume of the urine is increasing, it exerts pressure on the walls of the bladder and they are stretched because it is expands. The walls of the urinary bladder are stretchable, expands, so that it exerts pressure on the walls of the bladder and it is stretched. The stretched bladder stimulates the nerve endings to develop reflexes. This triggers off an automatic reflex action which causes the upper sphincter opened. But still the lower sphincter is under the control of brain, that is in our control, and urine is still retained in the bladder until the second sphincter is relaxed. So the first sphincter relaxed when it is stretched by the walls of the urinary bladder. So because of the reflex action, it gets opened, but the second sphincter is in our control. Urge for micturition occurs when urinary bladder is filled with 300 to 400 ml of urine. But urine can be retained in the bladder till it gets filled up to the maximum capacity of 700 to 800 ml. But at this time, the urge becomes painful. If the bladder is filled up to 700 to 800, then it is very painful and it leads to the voluntary micturition. But 300 to 400 ml of urine is there, that time we feel the sensation. Generally per day, a person excretes 1.6 to 1.8 liters of urine, but its quantity increases or decreases. It depends upon the intake of the water or fruit juices, etc., etc. It becomes on the intake of the liquids. If the person takes more intake of liquids, automatically he excretes more. Otherwise, it decreases. But generally, 1.6 to 1.8 liters of urine is generally excreted from a human being per a day. This is all about micturition. So, let's summarize today's topics. Today we have learnt about the mechanism of urine formation and about micturition. That's all for today. Meet you in our next session. Thank you.